I love biology more than some people love their children.
Shut up, Haley.
Point proven. Anyways, my favorite process, which takes place in my favorite part of the cell, the mitochondria, also known as the powerhouse of the cell, is, can you guess it?
No.
Cellular respiration. That is exactly what we will be learning about today.
You have eaten glucose before. You're probably eating some right now.
Hey.
Yeah. Well, cellular respiration is the cellular process of oxidizing glucose to obtain,
Adenosine triphosphate.
thank you, also known as ATP, which is basically a fancy deluxe Visa Gold card for the cell.
I want one of those. Oh wait, I have them inside my cell. Okay, cool.
Okay. To understand this process, we have to understand some other key processes.
The first of these are oxidation and reduction. Hold my closer. Got it, okay. If we take a look at the formula for cellular respiration, we can see these in action. Oxidation is when a reactant is losing electrons and consequently energy. In our case, the glucose, C6H12O6, is losing one hydrogen, which is losing an electron, considering hydrogen is only one electron. The glucose is being broken apart into carbon dioxide, 6CO2. Reduction is the exact opposite. This is when a reactant is gaining an electron and consequently gaining a hydrogen with one electron. In our reaction, the oxygen, 6O2, is being reduced into water, 6H2O. This term reduction can be confusing This term reduction can be confusing due to the root being reduced. However, since you are gaining an electron, you're gaining a negative charge. Think of reduction as adding a negative or reducing the charge of the glucose. I'm done.
Another thing you should know are the two catabolic pathways. One of these being fermentation.
Yes! Really love fermentation. It's what yeast does.
One of these is fermentation, while the other is aerobic respiration. Fermentation basically means cellular respiration without oxygen, producing lactic acid or ethanol.
So if I hold my breath, can I do fermentation?
I'll tell you how you can do fermentation. This happens in your body during a time where oxygen is limited, such as when you work out.
Hey, I work out.
Okay, yeah.
So I do fermentation.
All right. Aerobic respiration is the opposite, which is cellular respiration where oxygen is present per normal.
Yeah. We all know the magical molecule ATP, adenosine triphosphate. Well, when ATP loses a phosphate, it becomes ADP, adenosine diphosphate. ATP loses a phosphate when a cell needs to use that phosphate for energy, such as during active transport.
The last background knowledge we need is that at key steps during cellular respiration, electrons are stripped from the glucose. Each electron travels with a proton, thereby forming that hydrogen molecule we were talking about.
Oh, I remember that.
Okay. The hydrogen atoms are passed to an electron carrier, the coenzyme NAD+. NAD+ accepts a hydrogen to form NADH, which we will discuss in cellular respiration. Let us now continue on with cellular respiration.
I happen to have a chart for the first step of cellular respiration.
She does.
Didn't just make it.
Cellular respiration begins with its first step called glycolysis.
Glycolysis.
Glycolysis occurs in the cytosol, which is in the cytoplasm. The breaking down of glucose begins when it is split into two pyruvate molecules. During glycolysis, two ATP molecules are consumed, which helps lead to stabilized glucose, making it more reactive. Later in glycolysis, 4 ATP molecules are produced. This results in the net gain of 2 ATP. 2 NADH are also produced, which will be used later on.
Most of the potential energy of the glucose molecules still resides in the remaining 2 pyruvates. After glycolysis, pyruvate is oxidized, which, if you remember, is when the electrons are removed. When the pyruvate is oxidized, it turns into acetyl-CoA. This step is sometimes referred to as step 1.5 because of the chemical change. During 1.5, a transport protein moves pyruvate from the cytosol into the matrix of the mitochondria. In the matrix, the enzyme speeds up 3 reactions. A carbon dioxide is removed to NADH, and coenzyme A joins with the remaining 2 carbon fragments to form acetyl-CoA. 2 acetyl-CoA molecules are produced per glucose. The acetyl-CoA now enters the citric acid cycle, thus concluding step 1.5. Next step is the electron transport chain. The electron transport chain is embedded to the inner protein of the mitochondria. It is composed of 3 transmembrane proteins that work as hydrogen pumps and 2 carrier molecules that transport electrons between hydrogen pumps. There are thousands of these within the mitochondrial membrane as hydrogens are moving from a high to low concentration constantly all the time. The electron transport chain is powered by electrons in the molecules NADH and FADH2. As the electrons flow through the electron chain, the loss of energy by the electron is used to power the pumping of protons across the inner membrane. At the end of the electron transport chain, the electrons combine with 2 hydrogen ions and oxygen to form, you guessed it, water. The oxygen sort of
love water so much.
Good for you. The oxygen sort of sit at the bottom of the membrane and catch the remaining hydrogens as they go through the pumps. Notice that oxygen is the final electron acceptor, and when it is not available, the transport of electrons comes to a screeching halt. No hydrogen atoms are pumped and no ATP is produced.
The hydrogen ions flow back down the gradient through a channel in the transmembrane protein known as ATP synthase.
ATP synthase, fun fact, makes ATP because of the word synthase. It says it speeds up the production of ATP.
Okay, this isn't your part of the video. ATP synthase harnesses the force of the hydrogen ions entering through to add a phosphorase to ADP, forming ATP.
I get it, like adenosine diphosphate to adenosine triphosphate because the di, which means 2, is added so it becomes 3, like tri.
Yeah, stick to your video. It's your part of the video. The proton motor force is in place because the inner membrane of mitochondria is impermeable to hydrogen ions. Like a room with one exit, electrons are held behind the inner membrane with their only available exit being ATP synthase.
This process is referred to as chemoosmosis.
Yeah, something smart about that? Yeah.
This combined with the electron transport chain composes oxidative phosphorylation. This specific term is used because ADP is phosphorylated. Because a phosphorus is added, an oxygen is necessary to keep the electrons flowing. 1 molecule of glucose will allow for 30 to 32 molecules of ATP to be produced. This means you can thank cellular respiration for all the energy you have throughout the day, or don't have because of the homework from VJA. Thank you for watching from both of us, and thank you Tracy Sicalo for being an awesome AP biology teacher all year.
Yeah.
And we hope you learned something about cellular respiration.
Yippity duck.
Do 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 do.
No.
That was too long.
What?
Hey. Damn. He's in action. Where are my posters? And where did they? Is losing. I have to restart. I'm gonna make sure.
Oxygen is
It's present.
while the other
Oh, I missed it. Go back.
The other thing you should know are the two
No, go back.
Okay. Cellular respiration.
This step is some. Oh my god, we had a chart for this.
We did.
Oh my.
Should I stop the video?
Yes.
Yeah.
Okay.
After glucolysis. Start again.
Thank you so much. Sorry.
Step 1.5. Stop the video.
Stop the video. Okay. In the citric acid.
I just can't believe you made your finger in your nose.
Oh, I can probably take that off. In the citric acid cycle, which
Okay,
is...
no, start now. Inner membrane of the mitochondria. It is composed of 3 transmembrane proteins that The next step is the electron transport chain. The electron transport chain is embedded in the inner membrane of the mitochondria.
I wasn't looking.
It is, I swear to God.